exit stage left. Thank you. Dr. Jaenisch, you want to sit down for a second? I have just a few welcoming remarks. Everyone in the stem cell world knows Dr. Rudolf Jaenisch, our science keynote speaker. He is a giant in the field. His work with transgenic mice, his work with stem cells has earned a Lasker Award, and we all are very honored to have him and see him as a national treasure. He was recognized last week by President Obama, as many of you know, with the National Medal of Science, the nation's highest scientific honor. But since we all know about him, I have to share a little story about Dr. Jaenisch, because when I became a full-time stem cell advocate and was deciding whether I wanted to leave a practice of law, a lucrative practice of law, and change my life 10 years ago, I spoke to two scientists. One was Dolly the sheep's father, Dr. Ian Wilmot, and the second was Dr. Jaenisch. And both of them encouraged me to consider supporting this cause. I had helped debunk a fraudulent or a claim, an outrageous scientific claim, and realized that there was a lot of forces out there that were impacting in a negative way this exciting field of stem cell research. And Dr. Jaenisch invited me to the very first meeting of the International Society of Stem Cell Research in Washington as his guest. And there I met the scientists that I saw were going to lead the world to the future of medicine. And I became very, very inspired. Dr. Jaenisch became the founding member of the Science Advisory Board for my organization, the Genetics Policy Institute, and I found by just being able to use his name that I was able to kick down doors that otherwise I would have never been able to do. So I'm deeply honored to present our keynote speaker, Dr. Rudolf Jaenisch, and thank him.
Well, thank you very much, Bernie, for this really nice introduction. I give you my story now, meeting Bernie. So I got this call from a lawyer, which I find is not necessarily what I was most interested in, but I was very impressed when he told me he would go after these clowns and charlatans who in the beginning, after Dolly was generated, were promoting human cloning and really muddling the field, and it was very complicated. And I thought, Bernie going after these people as a lawyer really put the whole discussion on a different level, and I think that was really important. So thank you for inviting me to this meeting. And so I would like to talk about, and I have the next first slide, about stem cells and how we think about this and what are the issues which we are concerned with. So the promise of stem cell research, of course, is tissue engineering. You can make all these cells, all cells of the body in the petri dish. And that, of course, has enormous potential. And the goal of stem cell research, as is defined by many in this field, is to use the potential of these embryonic stem cells, or pluripotent cells, to provide matched cells for customized tissue repair. Now, the concept of personalized medicine would be to use your own cells, so you wouldn't have to deal with transplantation, rejection, and so on. Now, when I grew up, I learned that development is unidirectional. It goes from the pluripotent cell to the differentiated cell. But what Dolly told us was you can reverse this process, which told us that the nucleus of a differentiated cell retains the potential to generate a whole organism. If you put together embryonic stem cells and nuclear transplantation, you come to the concept of therapeutic cloning. And that was much discussed in the end of the 90s. And indeed, it was important to ascertain that this would be a possibility that really works. So what we did was to make a proof of principle experiment where we use as a patient, if you want, a mutant mouse which has no peripheral B and T cells, and then took a skin cell from those cells, did nuclear transfer, generated a cloned embryonic stem cell, corrected the genetic defect, and then differentiated these cells to a blood stem cell, put them back into the mice, and they restore the immune system. So therapeutic cloning works. However, in mammals. However, when you replace the mouse with a human, this is just not an option because of many reasons, because nuclear transfer in humans really has not worked yet sufficiently to make it possible, and then also there are many, many objections. So the only the only alternative was to do it without human eggs. So and this was, of course, then the birth of the iPS cells in this landmark paper from Shinya Yamanaka now 5 years ago, which I think changed the field. So the Yamanaka approach was based on understanding what is the circuitry of embryonic stem cells. So what controls pluripotency, and these are these three famous transcription factors, OCT4, NANOC, and SOX2, which control all the downstream genes, and these genes also form a regulatory network where they influence each other's expression. So if I just put the Yamanaka approach into one slide, then it's amazing. You have a cell growing in a culture dish, a fibroblast. You take a single cell and transduce them. That means insert these genes, SOX2, OCT4, KLF4, and c-Myc, two oncogenes, and what you get out is a mouse. So this is really, as I said before, a single skin cell can be induced to organize a whole complex animal, which is really quite amazing as nuclear transfer, I think, and that changed the field. So what we understand about the mechanism of this, and I will just summarize this in one slide, is that this direct reprogramming is reestablishing the core regulatory loop, autoregulatory loop of embryonic stem cells. We have found that every somatic cell has the potential to generate an iPS cell, but in the process initiated by these transcription factors, which are named, and it involves extensive epigenetic resetting of the genome and involves multiple cell division. So it's a long process, and I'm not going to talk about what we understand, but I just summarize it here, that the key is to reestablish this autoregulatory loop, and then all the downstream genes will fall into place. So, what would be the goal of human iPS and embryonic stem cell research? The goal would be to study human diseases in the petri dish. I think that is the most immediate and most exciting application. Then to use this to screen for drugs which might help to treat diseases if you can see the disease footprint in the petri dish. And eventually to go for customized cell therapy, although I believe this is not around the corner. But there are a number of unresolved complicating issues, and I want to come to those. And I think one of the key ones is, we often forget, human embryonic stem cells or iPS cells are not like mouse ES cells. With mouse ES cells, you can do anything. With humans, you cannot. They grow slowly, they grow single cells, and the single cell cloning is very inefficient. The cells, it's very laborious to work with these cells. They need to be passaged mechanically and not by trypsin, as it's in most ES cells, and they have a very low efficiency of homologous recombination. So it raises the question, what is a good ES and what is a good iPS cell? So well, let me address these questions a little bit. So the human embryonic stem cells were generated by Jamie Thompson in this landmark paper in the 90s, where he used cultured the cells in FGF and activin, but not in LIV, which is really key for culturing mostly embryonic stem cells. There are differences, as I said, between individual human yeast cells. So you have human yeast cell number 1 and number 2. They differ. They differ in their biological potential. And the quality of, and that's really, I think, a very important point, the quality of the yeast cells can be determined by the genetic background. They always come from different embryos. And by the culture conditions, how you isolated those cells, how you propagated them. And we were interested in defining some of these parameters. It really raises the question, what is a ground state of pluripotency? And is it realized in our classical human yeast cells as defined in the Jamie-Thomson protocol? So what we wanted to do is to change in a genetically identical cell some environmental conditions. In this case was oxygen tension. As you know, all our cells grown in standard tissue culture conditions are grown under atmospheric oxygen, 20%, which is cells in the body never see. They're always under oxidative stress. So we wanted to see, does this affect the quality of the cells? So we took human eggs and grew them in low oxygen, physiological oxygen. At the first passage, getting an yeast cell, they were split, and one part was continued to be cultured under 5% oxygen, and the other one under atmospheric oxygen. And this was then done for a year or two, and we asked the question, are these cells different? Are they of different epigenetic state? They're genetically identical. They're coming from the same embryo. And this was done by Maya Mitalipova and Chris Langler. So let me come to one important issue which we saw. That was X inactivation. You know, in female cells, one X gets inactivated, and it's known that most embryonic stem cells are pre-X inactivation. Both X's are active. But human cells, curiously enough, and really unexplained, they're post-X inactivation. One X already has undergone the X inactivation process. So we asked the question, is that process affected by the isolation procedure? And X inactivation occurs by activating the exist gene, which is on the inactive X, which encodes a non-coding RNA, which codes the whole X chromosome and converts it to a bar body. And you can see this in undifferentiated cells. Both X's are very low expressed. It's a fish experiment. But after differentiation, you see that the bar body is highly lighting up with exist, coats the whole chromosome, whereas the active X you don't see anymore because exist is turned off. So that's a decent test. So when we did that, you might be able to see this here, in the cells which were shifted up to 20%, you see every cell in the undifferentiated state expresses exist as this red spot. And this doesn't change when you differentiate these cells. So this is exactly like in conventional human yeast cells. But when the cells never saw high oxygen, there's no exist expression in the undifferentiated cells, but as soon as you differentiate them, they inactivate the X. That's exactly like in most yeast cells. So they're in a different state when you treat these cells with low oxygen. And what you conclude from this is that the epigenetic state, and the X activation will be only one parameter, is irreversibly affected by early environmental conditions, in this case, high oxygen. So when you shift the oxygen once up, you will be irreversibly inactivate the X chromosome. So, asking the question again, what is the ground state? What do we have to strive at to get iPS cells, to get pluripotent cells? Is it the conventional way, or is it low oxygen, or is it something else? Let me just come to an important issue. You can generate pluripotent cells from different stage mouse embryos. You can generate them from the inner cell mass of the blastocyst or from the implanted embryo, the epiblast. These are called epi-SC cells. And they're both pluripotent by many criteria, but actually they're very different. And I want to come to this difference because I think that's very informative. So this would be mouse embryonic stem cells derived from the blastocyst, or iPS cells from somatic cells, or cells, epi-SCs, derived from the implanted embryo, from the epiblast. In human, yeast cells are derived from blastocysts and iPS cells from somatic cells. But these cells are very, very different. The mouse cells are small cells, dense colonies. iPSC cells and human ES cells are flat colonies, large cells. They have different growth requirements, different cytokines, LIF versus basic FGF. Gene targeting is very efficient here, very inefficient in those cells. Exon activation, they are pre-exon activation, those are post-exon activation. And these states of pluripotency, they're different states of pluripotency, have been named by Austin Smith. The naive state versus a prime state. So the mouse iPSC cells really resemble conventional human ES cells, and they're very different from the naive classical mouse ES cells. So, what I want to say is these are two different states. Mouse naive cells are different from the human primed ES cells, which resemble these mouse iPSC cells. Can we convert them? And indeed, we can. By culturing the cells in LIF, and they convert to this state, or in FGF, they convert to this state. It's all done in the mouse, and is based on really important work from Austin Smith's laboratory. Can you do this with human cells? Obviously, we would like to make human naive cells. And it didn't work that well. So what was done was, in my lab, was done by what was this now? Okay, it was done by Jakob Hanna. I skipped over it. And he used a system which we thought was quite interesting and useful. It was to take human fibroblasts which carry all the reprogramming factors in a drug-inducible way. And we can just give the drug, doxycycline, tetracycline in this case, and generate what we call primary iPS cells. And then we can differentiate these cells and to fibroblasts. They're genetically homogeneous, and now we just have to only to give the drug to activate and generate new iPS cells. So you don't need novel viral infection. It's just a technical issue which I just want to point out. Now, using the system, we found that when you activate these viruses and add small inhibitors from Austin Smith, we can convert these cells, which are very flat, to something which looks like a mouse ES cells within a few passages. So this was very exciting. So the question was, what were these cells? Were they really like mouse ES cells? And I give you just X activations reminding you, in human ES cells, they're post-exon activation, but in mouse, they're pre-exon activation. And when you look at this, then you find that indeed these cells before treatment exist in every cell, but after treatment, none of the cells exist, and they reactivate the exist after differentiation. So every cell expresses no exist. So what it really says is that these cells, this conversion has reactivated the inactive X, and therefore is an important parameter for having human naive type of cells. So I will not go into this here, but rather come to the conclusion here. Why do we think this is important? So the most, we can make a nice and extensive comparison between the naive state and the prime state. They proliferate better, the naive cells, higher. They have a high cloning efficiency. Gene targeting is very high, low here. Pre-exon activation, post-exon activation. And these cells can generate chimeras, which is a very important test for pluripotency, whereas these cells cannot. So when you look now in the human cells, in those cells which are conventional cells, compare them with these converted naive cells, then you find that many of these parameters which we see in the mouse are now also in the human. So, for example, they proliferate faster, they have a high cloning efficiency, we don't know about gene targeting, they reactivated the X, and of course we don't know whether they make chimeras. So the key issue here is, or why we're interested is, does this naive state, if you can freeze it, if you can really maintain it, does it help us to manipulate the human ES cells in a way we do it with mouse ES cells? And would it facilitate working with these cells in our quest to study human diseases? So let me come to another very important issue, which is now much in the literature, which are differences between ES cells and ES cells, between ES cells and iPS cells, and between iPS cells and the iPS cells. Why is that important? If you study human disease and you have a patient-derived cell versus a control cell, what is your control cell? if the S cells or the iPS cells differ anyway, it muddles up your analysis, because your difference you might see might be not due to the disease, but due to the system immanent problems. And there are many, many papers have come out recently which document this, epigenetic differences between iPS cells and ES cells and others. So there are differences which I think, as I said, is a problem for using the system for human disease work. What could the difference be due to? Well, they can be due to the vector integrations, to expression, or the stoichiometry of the vectors, how are they expressed, and to the genetic background of the cells. And the conditions how you isolate the cells. For example, choice of inhibitors, high versus low oxygen, what have you. None of these things have been really carefully analyzed. So I will just come to the stoichiometry of factors. So these are the transcription factors introduced into the cells to induce iPS cells. There has been a very interesting paper from Konrad Rehlinger last year, which showed, and he was a former student in my laboratory, he showed that when you that the imprinting status of a given locus really affects the quality of the iPS cells. And he made really three main conclusions. The silencing of this imprinted locus, it's DLK1, DO3 locus, an awful name, really is lost in iPS cells, but not in ES cells. And the loss of this imprinting correlates with loss of pluripotency as measured by various assays. And none of these cells could generate what we call all iPS mice. I will tell you in a minute what this is. Now, the experimental system used was extremely well defined, and that's why this is a very interesting paper. What he used was the secondary system where the mouse carried these vectors integrated, and you just could now isolate cells, activate them with a drug, and generate iPS cells. And these iPS cells could then be tested in either chimera formation, you inject them in a blastocyst to make a chimera, or in what we call tetraploid complementation, which is an assay where you inject the cells not in a normal blastocyst, but in a blastocyst which is tetraploid. And these cells, the host cells cannot contribute to the embryo, but only the embryos will be only composed of these iPS cells, which is the most stringent assay of pluripotency. So these cells he gets are isogenic, they're identical genetically, and they have identical stoichiometry of factors. Now, Konrad came from my laboratory, and of course he has developed the system, this inducible system, and we worked also with the system, and another student, Bryce Carey, worked with this together with Stella Makulaki, and he used the same system, but he came to very different results. I will just contrast these two results, because I think they give us a very important message. So he used essentially the same system with very minor difference. I will come to these minor differences. And again, the cells he made were isogenic, and they have an identical stoichiometry. So all the induction was all identical. And so I'm not going to go through these results much more, but what you find is that, and I just want to come to the conclusion here, that in his system there is no loss of imprinting to the extent as found in the Konrad system, Konrad-Rehlinger system. And in addition, what you can see here, the quality of chimeras, which was rather low in the Konrad-Rehlinger's mice, in those cells which lost imprinting, there's no difference in our system. So there were major differences in how to measure pluripotency. And I don't want to dwell too much on this, on these results. I want to come to the conclusions. And again, I think I just leave this off here. All the cells we used could make tetraploid complementation. So there were major difference between these two systems. And I will not really go into these details here. And one other important thing was the mice he made all get tumors in a very, very short while, 2 months. All mice never got tumors. So, two almost identical systems, very different outcome. What is a lesson here? And that's what I'm coming at. So there's high tumor incidence, short survival in the KH, Kornato-Ehlinger mice, and no tumors in our mice. So what is the reason for this difference? Can we conclude something which tells us something about iPS cells? And that's what I want to get into. It's all control. So the major differences were very minor. This you see it's summarized here. So in his mice, the loss of imprinting was in 95%. In ours, retaining was only 30%, was 70% retained the imprinting. Tumor incidence was 100%, none. But everything else was identical. So what was the difference? Turns out the difference was very minor. And I just go into this. The vector design was different. There were 3 minor differences. These are the 4 vectors. The sequence, KS and SK, third and the second were different. Different control elements, I don't want to go into the details here, were used in these vectors. So for this audience, I will not really go what the differences are. I tell you they were minor in the totally unexpected for us. So those were the differences. And what we find was that the expression level of the protein was enormously different. And I just show you this here on this slide. So the key factor OCT4 in our cells, although the RNA was identical, was 10-fold higher and KLF4 was 20-fold higher, whereas the other 2 in our case were lower, this oncogene here. And this made the whole difference between those. And I want to come to the conclusion here, what we conclude from this. And I want to leave this out here because I think it's... So what we did was, in Conrad's experiment, it's a very complicated experiment, he isolated his cells, and the conclusion we draw is when you overexpress, when you increase the protein of OCT4 and KLF4, 2 of these transcription factors, you affect imprinting. And that's what we can see here. And I show you this in a very complicated experiment, which I don't want to go into, where we derive these cells one after going into deriving them, getting a clone, putting the gain in a mouse, and then deriving a clone, putting a gain in the mouse, and getting these tertiary cells. And what we find is that the imprinting in the original cells was 5%, then it was 20%, we come to 60%. So we can fix the problem, basically. We can fix the problem of the low quality of iPS cells by increasing the stoichiometry. So what are the conclusions? This is here. What we learned is that a high-quality iPS cells requires high OCT4, KLF4 expression. They can both make teratomas. The chimera grade is high here, it's low here. The tetraploid competency is high here, very low here, and they are mostly normal imprinting. So what does affect the abuse state and the biological characteristics of individual iPS cells? It's a genetic background. It's conditions that are particularly for a given iPS, such as a vector integration, which is of course identical in the 2 mouse strains, the factor expression level, and the stoichiometry in the initial phase of reprogramming. That's the only difference in these 2 mouse strains. So basically, because we compare 2 very well-defined mouse strains, we can make these conclusions. Now, what does it mean really for the issues we have to consider when we think about good iPS cells? Can we define really a generic state of iPS cells, or is rather the state of an individual iPS cell defined by idiosyncratic conditions of its derivation? And can we optimize it? I think that's another really important point. So when we think about the application here of this whole approach, it's for disease modeling and eventually for cell therapy, then we will have to realize the potential. We have to resolve a number of issues. One is gene targeting, and one is to get a disease-relevant phenotype. And I'm running out of time, so I will really just not go into much detail anymore. We have generated a new way to target genes, which is very key, which uses zinc finger nucleases and tau. I will not go into this anymore. It is really a novel way of inserting genes. into the human iPS cells, spearheaded by these two individuals. And what we can do now is to target genes at will, which we have now probably a dozen genes which mix with an efficiency as we can do it in mouse ES cells. And I'm sort of running out of time, so I will really skip over some of these key new developments and just come to, sorry about this, come to disease phenotypes in my last few slides. So far, iPS technology has been used to find phenotypes in a number of examples, in Rett syndrome, spinal atrophy, progeria, schizophrenia, and some heart muscle diseases. We have been interested in Parkinson's, and we have failed to find a phenotype in sporadic mutation, also in sporadic examples from iPS has derived from sporadic Parkinson patients. Although there are no reports coming up where there's some really becoming interesting science. But there's really one key issue which I want to make you aware of. Parkinson develops over decades. But when we make an iPS cells, we don't have decades. We have maybe weeks or months to do this. So we have to understand how to differentiate the cells through dopaminergic neurons, and then possibly expose these cells to stress situations which are thought to contribute to the disease, and maybe telescope the aging process to see a phenotype. And the key challenge, I think, to my opinion, is when you do these experiments, do you have a right control cell? What is the cell which you can use as a control when you see you have a phenotype which is disease relevant? And I said before, the difference between iPS cells and iPS cells makes this an important issue. And even a healthy sibling might be not a good control because it's genetically different. So our key issue was, can we make isogenic controls which differ only at the one base pair which leads to the disease? So generate either introducing a mutation, a single mutation, correcting mutation in a iPS cells, or introducing this point mutation into normal ES cells and generating isogenic ES cells. And really, I want to give you just my last data slide here. This is synuclein, which has many point mutations, which leads to early onset Parkinson. And what we did was to take two of these mutations, this mutation, a point mutation, this mutation, and generated basically cells which exclusively differ at this one base pair. So either at the AT53 mutation or the AK46 mutation. And we have different targeting strategies, and I will just not go into this anymore because I think we're running out of time. I want to say that we have now these isogenic controls which differ exclusively at this one base pair. And if we find now a phenotype in the culture dish, we can be reasonably assured this is disease relevant. So I think that's another very important tool which we'll have to use in the field. So on my last slide, clearly what the challenges are, we have to remove the vectors. We need equality of pluripotency, naive versus primed iPS cells, low versus high oxygen, what is really the best cell type we can use for disease research. Efficient genetic manipulation is important. And then, of course, a differentiation to phenotype. So I believe that the scenario, which is outlined here, where you take a patient, you have a patient in the hospital, you derive iPS cells, maybe routinely at some point, will eventually lead to study the mechanism of complex human diseases, maybe to find the effect of drugs, and eventually for cell therapy. So I want to summarize, just acknowledge the people in my lab who did the work, particularly students, Bryce Curry, Stella Makulaki, Frank Solman, Dirk Hockemeyer, Chris Saha, and we collaborated with Harvard people, Ginny Lee's laboratory, and with Sangamo. Thank you very much for your attention. So in case there are any questions, we have
Do you know if low oxygen works through ERK, STAT3, or GSK3 pathway to activate the X chromosome in stem cells?
So the question was, do we know the mechanism of high or low oxygen? I think it's just a stress response. So these cells are grown under the conventional Jamie Thomson conditions. Even when you freeze them, they tend to inactivate, or many clones do inactivate. So I think you expose the cells to stress, and the stress response is, among other things, I think X inactivation. Whereas I should say these naive cells, they're all grown under 20% oxygen, and they reactivate the X chromosome. So that's a different state of pluripotency.
Okay. Hi, thank you for the excellent talk. Have you tried to derive human ES cells with those with small molecules instead of FGF? Yes. And can we get naive human ES cells?
I think that's one of the key experiments. I think that would be, to my opinion, would be the key remaining experiment. Can we really derive, like in the mouse, right, the naive state from human embryos? We are not there yet. So at the moment we need transgenes, and that's difficult. So we have to define those conditions, and I think, to me, that's one of the most important experiments.
But just so that I don't waste my embryos in time, have you tried it and it doesn't work?
We have tried, it doesn't work with the conditions we had defined, so we have to get more sophisticated.
Okay, thank you.